The journal article that our group has chosen is the bioremediation of oily seawater by bacteria immobilized in biofilms coating macroalgae. For introduction, few research stated that bioremediation method is very practical for cost, but not for the open sea in treating oily seawater. This is because coastal materials can accommodate more hydrocarbon utilizing bacteria compared to water body. In other words, coasts have higher potential for oil biodegradation. However, the major issue with open sea is that any seeded microorganisms or added fertilizers will be washed away and diluted. Hence, seeding with immobilized microorganisms can be a solution. Through this research, they also found out that macroalgae that is collected from intertidal and subtidal zones of Arabian Gulf are richer in hydrocarbon utilizing microorganisms compared to water sample. Those microorganisms were found immobilized in biofilms coating the tali. The objective of this study is that to report on this phenomenon and also to test the potential of those naturally immobilized microorganisms for bioremediating seawater artificially polluted with oil.
The methodology of this research consists of sampling, microbial analysis, hydrocarbon utilization potential, and electron microscopy. Firstly, 10 macroalgae samples were collected. They are from the genus of Chlorophyta, Phaeophyta, and Rhodophyta. They represent predominant macroalgae in the Arabian Gulf. After sampling, microbial analysis was done. The crude oil utilizing bacteria were counted using the standard plate method. After being counted, the representative strains were isolated, purified, and identified. And then, the strains were compared with the strains that had been previously identified. The third method that was done was the hydrocarbon utilization potential. The potential of microorganisms associated with macroalgae for hydrocarbon attenuation in seawater was determined quantitatively by liquid gas chromatography. The final procedure in this research was the electron microscopy. For scanning electron microscopy, the materials were dehydrated in an ascending concentration series of acetone, which are 30% to 100% concentration. The dry samples were coated with gold before finally being examined.
Results for this experiment shows that 1 gram of microalgae records the reaction of 30 million cells of oil-utilizing bacteria. The amount of bacteria were enhanced in 2 weeks, being shaken in hydrocarbon-containing seawater. Overall, no cardioforms were recorded amplified more than Acinetobacter after 2 weeks. Observation under the electron micrograph showed rod-like filamentous nocardioform fragmenting perpendicularly to the microalgae surfaces. Hydrocarbon as the sole sources of carbon and energy for the oil-utilizing bacteria were compared with the nutrient agar. Result shows that the growth of nocardioforms isolates having good growth on aromatics benzene, including benzene, phenanthrene, naphthalene, xylene, toluene, pyrene, and biphenyl, and also shows good growth on N-alkanes, carbon number 9 to carbon 12, while on carbon 13 to carbon 30, carbon 32, carbon 36, and carbon 40 recorded excellent growth. While for Acinetobacter, good growth were recorded on aromatic benzene and N-alkanes of carbon 9 to carbon 11, carbon 24 to carbon 30, while carbon 12 to carbon 23 shows excellent growth of Acinetobacter. In all cases, hydrocarbon were recorded reduced in amount, and the oil-utilizing bacteria increased by 32 to 490-fold, immobilizing the macroalgae. Hence, the increased amount of bacteria is correlated with the reduction of hydrocarbon. The analysis of the seawater batches shows only 1 to few thousands of cells per gram of algae per milliliter of water were released into the water. Predominantly, the bacteria identified are Nocardioforms and Acinetobacter.
Next is discussion. Based on the result, phytoremediation by a hydrocarbon utilizers that is associated with macroalgae could be used as a solution for oily sea waters. Based on the result, macroalgae is rich in consortia of oil-utilizing microorganisms which were found on the surface of the macroalgae. Next, bacteria that is adhered to macroalgae are capable to reduce hydrocarbon concentrations in seawater. Even though only one aliphatic and aromatic hydrocarbon were investigated in the study, predominant microbes, Nocardioforms and Acinetobacter, could utilize many other aliphatic and aromatic hydrocarbon as sources of carbon as well as energy. Based on the result, it showed that the microbes were capable to attack almost all hydrocarbon fractions of crude oil. Besides, the study demonstrated the hydrocarbon attenuation in the seawater was due to the fact of the presence of the macroalgae. Based on the result, Nocardioforms contributed more in the process of attenuation of hydrocarbon rather than by the genus of Acinetobacter. Also, the result on the table showed that Nocardioforms isolates can live on a wider range of individual hydrocarbons as sources of carbon and energy rather than the isolates of Acinetobacter. In the result, bacteria, Nocardioforms and genus of Acinetobacter that are found on the surface of macroalgae, this is known as association. This association gives an interesting feature between these two organisms, which is its immobilization. In addition to that, the association between these two gives benefit to the microorganisms in which it may provide 1. Oxygen, 2. Nitrogenous and phosphorus compound, and 3. Vitamins.
Immobilized bacteria are highly potential to clean up the oil contamination and can aid in oil biodegradation in polluted environment. In this study, it was concluded that macroalgae submerged in the seawaters are coated with biofilms rich in oil-utilizing bacteria that contribute to hydrocarbon attenuation in water. These natural biological consortia represent valuable tools that are highly potential for phytoremediation of oily seawater. Immobilized microbial technology is favored for its high cell concentration and low microbial loss. The microbiological analysis of the seawater batches at the end of the above experiment showed that only small numbers of oil-utilizing bacteria were released into the water. Next, the individual bacterial isolates could grow on a wide range of pure alkanes and aromatic hydrocarbons as the sole sources of carbon and energy. This study also offers evidence that bacteria adhering to macroalgae are capable of substantially reducing hydrocarbon concentrations in seawater, and these immobilized microorganisms can potentially attack almost all hydrocarbon fractions of crude oil. However, there are also some limitations. Bioremediation approaches such as seeding with hydrocarbon-utilizing microorganisms and fertilization with nutrients may be technically feasible for the coast, but obviously not for the open sea. Next, oxygen concentration also has long been realized as one of the limiting factors for microbial hydrocarbon degradation in aquatic and terrestrial environments. Lastly, it is important to establish procedures and install control measures to prevent the oil spillage in the ocean, especially at the open sea. Implementation of the chemical and biological treatments also can help to solve the spillage problems. For example, by using the absorbents in cleaning up the oil spills.